This is our experiment on the effects of magnesium sulfate on the expression of the gene HSP150 in Saccharomyces cerevisiae by Aaron Bloom and Mia Bradley.
We're testing the effects of HSP150, a heat shock gene found in Saccharomyces cerevisiae, and what happens to it when exposed to magnesium sulfate.
The purpose of this experiment was to test the effect on the expression of HSP150 with different amounts of magnesium sulfate in Saccharomyces cerevisiae.
Our hypothesis was that the higher concentration of magnesium sulfate, the lower the expression of HSP150 in Saccharomyces cerevisiae. The major steps of our experiment was to create the OSC of yeast, add the varying concentrations of magnesium sulfate to yeast cultures, leaving a control with no magnesium sulfate, extract RNA and convert it to cDNA, and once it's turned into cDNA, run a real-time PCR test and analyze the data. We created a control yeast culture with no magnesium sulfate, a yeast culture with 10% magnesium sulfate, a yeast culture with 20% magnesium sulfate, a yeast control with 30% magnesium sulfate, a yeast culture with 40% magnesium sulfate, and a yeast culture with 50% magnesium sulfate.
The materials needed for this experiment is 50 milliliter conicals, magnesium sulfate, YPD broth, Saccharomyces cerevisiae culture, serological pipette, and orbital shaker.
Methods and procedures. Make sure to use sterile technique at all times throughout the procedure. First, set up an OSC to grow overnight. 24 hours is mandatory before adding any add-ons. Then, prepare the magnesium sulfate in the desired concentrations and set up YPD broth. Then, put the yeast cultures into conicals, labeling the different concentration levels of magnesium sulfate. Samples are taken and put into 1.7 microliter EPIs. RNA is then extracted and converted into cDNA. Once you have the cDNA, you can run it on a real-time PCR machine, making sure to set up your proper run. Then, analyze the results and record the data.
After getting our results from the real-time PCR machine, we used t-tests to figure out more about our results.
By looking at our data, we can tell that there is no clear trend on the effects of HSP150 when introduced with magnesium sulfate.
By looking at the graphs, it is clear that there is no real trend. Also, some of our results were not very accurate. Because of this, our hypothesis was not correct.
Acknowledgements. We got this idea from our teacher Henry Weeks, who has worked with yeast in the past. After doing some research, we decided to look into Saccharomyces cerevisiae, one of the most widely used types of yeast. While searching for research articles, we stumbled upon a certain gene called HSP150. HSP is a heat shock gene, which we found can be manipulated by magnesium sulfate. We decided to test the effect on different concentrations of HSP150 of the Saccharomyces cerevisiae. The testing and lab work was done at WF West High School in the cell culture laboratory.